Okay, we saw a brief introduction to how plants are responding and how much we know and how much we are trying to know and how difficult it is to understand them. Just imagine the scale of this problem. The ecosystems, biospheres are very diverse. Even a little patch of land can have so many little things growing, and if you allow nature to recover, it creates quite a complicated ecosystem, especially in the tropics where you have temperatures and precipitation, humidity that may be favorable unless you are in the deserts. But trying to understand how each species may be responding, or the same species may be responding at different locations, versus how it may be responding based on its altitude, or how it may be responding at a molecular level, chemically and genetically, through leaf morphology changes, etc., etc. It's an incredibly complicated problem. This article is a review article on all those aspects, and it's a very nice, very long paper with lots of beautiful references. You want to read this if this topic is of interest to you, or if you're pursuing research in this direction, because it's incredibly detailed and lists all the things we know and the challenges we face. So I'm obviously going to be brief about this. So we'll read the discussion, which is nice and long, which summarizes the main ideas again. So we'll focus on that, but look at some figures. And this is called Review of Scaling Plant Responses to Heat from Molecules to the Biosphere. So literally things happen at molecular level, like synthesizing proteins or fatty acids and so on, to biosphere level. You can think of maybe biosphere is optimizing something like the health of the biosphere, its complexity, its resilience to shocks, and overall its productivity. These are ideas. We don't know exactly if that's what happens, but predicting plant responses to rising temperatures, including actual heat waves and hot droughts of varying intensity and duration, is central to addressing the climate and biodiversity crises. However, plant responses to heat are scale dependent, complicating cross-scale predictions. You ultimately want to understand where we are headed with this, so you want to be able to predict the impacts as warming continues with extreme events and so on. We highlight recent progress revealing how and why plant responses to heat change across scales, including scales of biological organization and space versus time. Turns out that spatial and temporal heating tend to have different responses, so maybe there is a species level response and a biosphere level response, or ecosystem level response. We give examples of scaling up from molecular and leaf scale data and processes which are modified by homeostatic and buffering mechanisms at whole plants and ecosystem scales. Homeostatic is where you are trying to stay the same by responding to changes, and buffering is basically you are finding ways to delay the response to changing temperatures, for example, or vapor. We show that scaling down predicting plant responses to warming from broad scale spatial patterns can also be misleading, even in direction. Is it going this way or exactly the other way, or in some angular fashion? Addressing such scale dependencies is essential to improving the prediction of plant responses to heat. Remember that I'm not really an ecologist. I've learned enough about the biological models, especially in the ocean and so on. So I know a bit, but this is about reading these and understanding them, reading all the related material, and getting enough knowledge to be able to translate that for you. You may be an expert who will read things, or maybe even suggest what I might have done, misstated something or so. But if you are like me, a novice, you will hopefully learn from this as well. So let's read some upfront material of each section. Scaling up from biochemical kinetics. This is the start of the sections. I've jumped the introduction, which sets the stage for how we are warming, how ecosystems are getting stressed, biodiversity losses, etc. Even fragmentation of forests, deforestation, sea level rise, salinification of soils, and so on. From first principles, biochemical reaction kinetics provide a strong foundation for predicting the effects of heat on plants. Acute heat exposure causes injury, reversible or irreversible cellular impairment, and necrosis, so irreversible cell death, both of which are largely driven by protein denaturation kinetics. So that's a very technical term that I will leave, but you can go ask ChatGPT or do the homework. Thus, rates of protein unfolding and cellular damage, so that's protein denaturation, protein unfolding, and basically they are all designed to function together with other chemicals like plastids and then proteins. There is a growth of the plant, there is procreation involved, there is maintenance, somatic and gonadic maintenance they call it, and so on. So proteins are involved in everything, so their response is going to matter a lot. Okay, so thus the rate of protein unfolding and cellular damage increase exponentially with temperature, and the time required to reach a given damage threshold, such as proportion of cellular processes or tissues affected, decreases exponentially with increasing temperature. Okay, time required to reach a given damage threshold decreases, which is bad news. Less severe heat stress and or shorter exposure times often injure photosynthetic machinery first, causing inactivation of rubisco, which is a critical enzyme, and impairment of photosynthesis, impairment of photosystem 2. These injuries have traditionally been quantified by using threshold temperatures that cause loss of function, with recent studies improving on this by characterizing responses as time temperature relationships. We discussed this very briefly in the previous podcast, so I won't go into details here, but either way I'm not going into that much detail. But photosynthetic heat tolerance can increase within hours of exposure to high temperature, driven by accumulation of heat shock proteins and metabolites that stabilize proteins. At higher temperatures and or longer exposure times, though, protein denaturation can exceed protective and repair capacities, resulting in irreversible necrosis or cell death. So I won't read in specific examples, but the seasonal timing of heat exposure also matters. At what stage of the life cycle of phenology the plant is can matter in terms of the impacts of heat on the plant itself. When extreme heat coincides with bud burst early in the growing season, it can reduce total leaf area and carbon assimilation for the remainder of the season. So one hit in the beginning, and then the entire season response can change. Okay. Secondly, scaling up from stomatal responses. Leaf level theory about stomatal responses do not always predict how. Water use and evaporative cooling vary across contexts or scales, complicating prediction of plant responses to heat. We discussed this just a little bit in the previous podcast. Water loss through stomata, which we call transpiration, plays a major role in regulating leaf temperature and limiting heat injury and necrosis under extreme conditions. Transpiration is also coupled to carbon assimilation because CO2 enters leaves through stomata as water is lost. Stomatal organization theory hypothesizes that plants regulate stomatal conductance to maximize carbon gain relative to either marginal water loss or marginal hydraulic cost of lifting water, right? You want to optimize stomatal conductance or the opening time so that you capture enough CO2 without losing too much water. Then there are details about warm temperatures, higher humidities, how are they affecting the stomatal conductances, and what to use versus dry hot weather, how is it changing vapor pressure deficit and stomatal functioning as well. Stomata are predicted to close at high temperatures, reducing transpiration to either maintain water use efficiency or minimize hydraulic risk. We'll look at a figure this. So however, emerging evidence shows that stomatal conductance sometimes increases at higher temperature even as photosynthesis declines. This suggests that transpiration may be prioritized for leaf cooling to avoid heat stress and necrosis, protecting carbon invested in leaves and enabling continued carbon gain rather than optimized for short-term carbon gain. So obviously you understand the complication that plants are trying to optimize not dying or dying of cells versus continued photosynthesis. So just survive and then keep alive, stay alive, and then continue once the heat is gone. What happens if the heat keeps on increasing and you cross a threshold? So those are key questions. Acute versus chronic changes. This may be particularly pronounced in plants with a stable water supply. So you can obviously have continued opening of stomata if vapor pressure deficit is not an issue. For example, well-watered poplar trees maintain leaf temperatures 3 to 5 degrees below air temperature during extreme heat waves, so they are happily losing water, and evaporative cooling is keeping them cooler because there is no water deficiency. Okay, variable responses to heat, aridity, and drought. So this is the opposite of having too much water. Predicting plant responses to temperature is further complicated by the great diversity of mechanisms and strategies that plants have evolved to maintain homeostasis. So stay close to your comfort zone, let's say. Strong correlations between heat, vapor pressure deficit, and drought make it difficult to disentangle their individual impacts. Air temperature and humidity together determine vapor pressure deficit, which drives evapotranspiration and hence affects plants both through water supply, so available soil water, and demand transpiration, right? Root depths matter, and all sorts of other factors are involved. Okay, so without going into details, during acute heat waves, leaf necrosis can occur even when soil water is not depleted, indicating that excess heat, not water limitation, is responsible for cell death. So you can, in other words, evaporative cooling mechanism may become ineffective if there is a serious water limitation, because then temperature is also increasing, so neither is working and necrosis happens. In the face of acute heat stress, some plants can adjust their heat tolerance in a matter of hours, though through biochemical responses, or buffer against spikes in leaf water potential caused by increases in VPD by using water stored in the stem, or mobilize non-structural carbon for osmoregulation and repairment of embolized tissue. So there are many mechanisms that are working, and we can only guess how they are working separately or together, and how they are working in response to just heat temperature rise versus vapor pressure deficit versus water availability, and so on. Over longer time scales, plants more commonly experience sustained periods of concurrent heat and drought lasting weeks or even years. So they have evolved to do that. So we are talking about two things. What have they evolved to do in any given climatic zone where they are existing, and what their responses will be as warming induced by our greenhouse gas increases are going to affect the plants, right? So very rapid growth versus slow evolutionary time scale growth are very different. And then there is a question of how good is evolution at responding to rapid changes. That's where we mentioned within hours they can adjust a few things by biochemical reactions. The two approaches to predicting plant responses to heat: scaling up and scaling down. So you are going from molecular level processes to cellular level, so leaf level gas exchange, etc. And you can see there are various things happening at molecular levels: protein synthesis, protecting proteins or other organelles, and so on. So if you want lagging processes, evolution and colonization, you want prediction going in this direction from data, homeostasis, emergent properties to prediction, versus scaling down from space for time substitution, with data looking at lagging processes such as evolution and colonization, and how to use that for making predictions for future responses. Here obviously evolution will be difficult, but people monitor, do lab experiments to understand what evolutionary times or genetic responses, let's say, happen. And colonization is basically if they move, displace themselves to survive in a cooler climate, let's say, or wetter climate, how does that work in terms of scaling down. Okay, so that's prediction here. Going to so there is the kinetics of photosynthesis, and scaling up, you are going from data to homeostasis to emergent properties, and then you want to predict. So there are multiple things. I already said this, but leaf morphology, thickness, stomatal conductance, number of stomata, fatty acids, various things can happen. Plants themselves respond, and then there is population, community, and ecosystem level response. Whether that will be based on the competition or cooperation or so on, we don't know that. Species distribution and biome level responses happen, and globally we consider it the biosphere of the earth. And is there something happening at that scale? How do they know that somebody here is responding and they have to respond in synchrony or as a competition? We don't know that, but maybe it's happening through climate or maybe some other mechanism that we don't yet know. Okay, so the second figure is on leaf and whole plant responses to excess heat. So I'm skipping the captions, as you can see, as is my usual habit, because if I can explain the figure. It paused me for some reason, and let me hope there is no discontinuity. I'll check and make sure. So we moved from figure 1 to figure 2, which has a nice long caption. As I was saying, I don't read the caption especially for descriptive type of schematics, so that I can just explain without getting into long captions, which are useful. You should read them for sure. So here, baseline conditions, always you want to understand the responses with respect to baseline conditions. Without that, you may be misinterpreting things as to what is the response and what is normal conditions, right? Normal behavior. Excessive heat conditions is the response we are looking for. Leaf, CO2 goes in, there is H2O loss, and then depending on whether the leaf temperature is below the critical temperature, or if leaf temperature is near the optimal, responses may include maximizing carbon assimilation, minimizing water loss, and leaf temperatures to stay below T critical to make sure the functioning goes on without necrosis or decreased photosynthetic efficiency, etc. And whole plant response, of course, CO2 is coming in, H2O is going out. Generally, we refer to it as evapotranspiration. Homeostasis balances carbon allocation, carbon used for respiration. So you photosynthesize, produce carbohydrate, and you burn it to generate energy, right? That's the goal. Osmoregulation, where the flow of fluxes across cells, whether it's CO2, water, nutrients, whatever, is regulated, and with osmotic pressure. Growth, repair, defense, reproduction, and exudation is organic material that is being exuded typically in the soil. So this is happening both in the atmosphere and in the roots and soils, right? So if there is excessive heat condition, then leaf necrosis can happen, as you can see here. So H2O loss may go up, so we are looking at CO2 changing the arrow size. So maybe more CO2 is available, or less CO2 is being taken up. So you may reach critical temperatures because you are not able to control anymore, and leaf temperatures have gone beyond optimal in one case. So this is response 1, where reduction in water loss or decreasing G3, so you can go and read what they are. Reduction in carbon assimilation, leaf temperature reaching T critical. And here in the second response, you are looking at still temperature remaining about optimal, but leaf temperature being below T critical. So increase in water loss, so increasing GS. Yeah, I should say it's GS. Reduction in carbon. I read these things and then it takes me a few days or few weeks to record these, and then anyway, you understand. Leaf temperature below T critical. So there are some schematics, but these are understood from various experiments and observations. Short-term responses for the tree then increase transpiration to cool, prioritize carbon for embolism repair, and increase the thermal heat tolerance, buffer water stress with stored water. And in the long term, change. So there are little changes in the arrows we have to pay attention to here. You are losing more water to increase transpiration. Here you are not. So compared to the control, for example, changes in leaf area and leaf angle, leaf shedding to reduce photosynthesis. They are like solar cells, but they are taking solar energy, producing carbohydrates. But if you have too many of them, you have to supply them resources like water and nutrients. Reduced biomass growth, changes in anatomical traits, investment in below ground biomass. So you can see the increase in below ground biomass here. So these are schematics of the responses. I will end up splitting this into 2 podcasts because it will get too long for just one podcast. Predicting the impact of heat, or actually let's do some clarification right here. Did I miss something? Okay, not quite. Okay, so let's go ahead. I'll clarify a few things in the next podcast if I missed something. Predicting the impact of heat or drought on plant productivity is further complicated because there can be conditions under which photosynthesis is decoupled from plant growth. So I'm taking bits of text that I consider are important for giving you an idea of what the review is. Plant growth, defined as cell division and expansion, is sensitive to changes in turgor pressure, which in turn is sensitive to water potential. In particular, it has been shown that tension at which cell expansion ceases is substantially less than the tension that induces stomatal closure and limits photosynthetic CO2 uptake. So these are very mechanistic levels on how plants work and cell divisions and cell expansions work. So you want to read more if you are interested in those details. So growth can be limited by environmental conditions that permit cell division and expansion. So that's called sink limited growth. By contrast, it is more commonly thought that plant growth is limited by availability of the carbohydrate products of the photosynthesis itself. It's called source limited growth as opposed to the sink limited growth here. Okay, this source limited paradigm is at the foundation of most global terrestrial vegetation models, the so-called dynamic vegetation models, or others that are simpler in some ways. There is evidence that in cases where plant growth is source limited, a reduction in photosynthesis in response to heat or drought does lead to a reduction in net primary productivity. However, under sink limitation, elevated temperatures and drought may lead to the cessation of growth well before the photosynthetic rates cease. So your plants are potentially optimizing this, but we don't know the details yet. Okay, now scaling up from leaves to canopies, then we went from leaves to a tree. If you go to the canopy level, there are emergent properties. So scaling plant productivity from leaves to landscapes also presents challenges owing to emergent properties of canopy structure and function. For example, leaf temperature, which is a key driver of carbon and water fluxes and thus grows primary productivity, is governed by an energy balance among radiation, convection and latent heat or transpiration fluxes. These fluxes are influenced by leaf traits such as size and stomatal conductance, but also by crown morphologies such as leaf angle and clumping, and canopy complexity as well, such as porosity and surface roughness. Together, these shape how radiation and airflow penetrate the canopy, influencing the vertical temperature profiles and leaf air temperature relationships. Very complicated. You can imagine even measuring these is going to be very hard, but without measuring you cannot really understand the processes or build models. To illustrate how canopy structure affects leaf and air temperatures, we consider radiation regimes and atmospheric coupling across 3 scales: solitary plants, moderately open canopies and closed canopies. So I'll skip further details, but you can go and read. They are very interesting. I loved reading the paper. I'm probably going to read it again because it's so dense that I don't always grasp the implication. I understand everything that's being said, but there are lots of implications of each point, which takes a long time to understand. Okay, microclimate buffering and leaf air temperature relationships in a tropical forest canopy. So let's look at temperature increase here and height. So you have air temperatures and then you have leaf responses in terms of height. Even in a climatology, you have changes in leaf structure, chlorophyll concentrations, etc., based on whether they are at the top with direct radiation and lot of light, or they are shaded by the other leaves and so on, because the concentration of chlorophyll determines how much you can photosynthesize, right? So thermal camera then at the canopy top, mid-story and understory. It's a complicated structure, just like clouds in the atmosphere. You have high clouds, mid clouds, low clouds, and they have all kinds of radiation properties and responses to warming, etc. So looking at time and photosynthesis, megathermy happens here, where photosynthesis drops and recovers over time. Poikilothermy, where you have steady response over time, whereas you have limited homeothermy, which means the response is to increase photosynthesis in the short term, but then it basically returns to previous levels, let's say. Okay, canopy top, where megathermy happens, leaf temperatures go up, and here mid-story, limited homeothermy, understory, limited homeothermy, but you can see the leaf temperature versus air temperature responses are very different. Here air temperature response is increasing leaf temperature, so either the photosynthetic limitation is not a concern, or the homeostasic method is, or process is not working, like evaporative cooling and so on. In the mid-story, you have much slower response to this is a sharper gradient versus this one. It is even less sharp, and this is somewhere here. So let's read this a bit more carefully. Microclimate buffering and leaf air temperature relationship in a tropical forest canopy. Typical midday vertical profiles of leaf and air temperatures. In this example, leaves can be up to 4 to 5 degrees cooler than air near the ground, and up to 7 to 8 degrees centigrade warmer than air at the canopy top. So that's what we were looking at here. So height versus temperatures, leaf temperatures, very much separated from air temperature, either cooler or warmer. Keep going in the wrong direction, sorry about that. Schematic representation of a generalized tropical forest canopy and field view of a thermal camera with the, why do you do this, canopy top, mid-story and understory. Okay. Illustrating the dominance of canopy top leaves in thermal imagery captured, so that's a data measuring problem. Hypothesized diurnal photosynthesis for leaves exhibiting different thermal responses. Limited homeothermy, light green, poikilothermy, gray, and megathermy, dark green. Okay, so you can see each response is shown here schematically. I already mentioned how it works. Okay, leaf versus air temperature relationships at different canopy heights, highlighting contrasting leaf thermal responses from limited homeothermy to megathermy. So intuitively you get a sense what it is, right? Homeothermy is where you are trying to keep the temperature about the same by responding and, you know, with cooling mechanisms, and megathermy is where you are warming, and poikilothermy is you are completely stable. So homeothermy responds with increased photosynthesis and then declines. Poikilothermy just is nailed flat over time, whereas megathermy photosynthesis drops and recovers. Okay, so did I miss something? That's okay. They are based on data from different tropical forest canopies. There are other details including biochemical kinetics and vapor pressure deficit. But even modest temperature differences can strongly influence plant and ecosystem functioning, owing to the exponential temperature sensitivities of biochemical kinetics within the plant leaves and vapor pressure deficit, especially when integrated over time. Relationships shown in A and D to F are based on data. Leaf temperatures were measured using some method, and I won't go into the details there. Okay, scaling down using spatial patterns to predict climate change impacts. This is the critical aspects. We are worrying about this basically because we are increasing greenhouse gases. There is a CO2 fertilization effect to worry about, but here is the vegetation responses or lack of responses to warming based on acute stressors like heat waves or chronic changes like temperature changes, and then duration, rate of warming, etc. matter as well. The use of spatial climate gradients to predict plant or ecosystem responses to changing climate would be valid if the processes responsible for spatial patterns were as rapid as the pace of changing climate. However, there is mounting evidence of contrasting plant responses to temperature variations across space versus time, suggesting that important ecological processes such as evolutionary adaptation or colonization of newly climatically suitable habitats, so shifting latitudes or altitudes, and these may lag the pace of climate change obviously. At the extreme, spatial versus temporal responses are opposite in sign. Okay, so spatial networks of tree ring data show that growth of widespread tree species in both eastern and western North America responds positively to spatial temperature variations. So trees grow faster at warmer locations, but negatively to time varying temperature. So they grow less in warmer than average years. There is a metabolic response in general in species where metabolism, it means growing may be faster at warmer locations, but negatively time varying temperature response, so they grow less in warmer than average years. So, tree rings capture season to season, year to year responses, and these are not necessarily year to year variations, are natural variabilities. So there is a response imprinted at that scale as well. Okay, so of course there are other details how space for time substitution leads to a prediction of increased tree growth for with warming, which is contradictory. So when you model, you have to know the difference in spatial versus temporal changes in temperature. Self-regulating mechanisms, I am jumping ahead, at the organism, population and ecosystem scales may help buffer transient negative impacts of warming, as discussed above, which I have skipped. Plastic response, which means flexible response, responses to heat or drought stress at intermediate time scales such as leaf shedding or increased biomass allocation to roots may buffer individuals from mortality, and the death of some individuals reduces density dependent competition for resources among the remaining individuals, potentially increasing their probability of persistence. So as an ecosystem, the whole thing may become more resilient if some of them die anyways, because then the competition for resources decreases with changing conditions. That's life, right? In the best case scenario, the intermediate time scales of these additional buffering mechanisms might bridge the temporal gap at longer time evolutionary and species range change, preventing the loss of biodiversity and ecosystem function. Thus, an important knowledge gap to fill is the time scale and limits of these buffering mechanisms from immediate to intermediate to long-term responses relative to one another, and especially relative to the pace of climate change. This will help better define which actions can be reliably addressed by using space for time substitution, and to what extent human interventions such as assisted evolution or assisted colonization may be needed to support species and ecosystem responses to climate change. Since an ecosystem may be responding and you don't have time to wait, you may just substitute time for space, because space is easier to deal with as changes are happening. Okay, so here is the some details in terms of the performance of vegetation's thermal performance. So this is the biogeography of plant responses to temperature variations. So here on this end, so this is colder temperatures and warmer temperatures. So temperature limited regimes, you have left skewed response, which means from the peak the tail is longer on the left side. So you can see the slope there. The slope will be very different, and the thermal performance curve here is, let's say, the normal response curve, and if you shift to cooler temperatures, you have left skewed response in terms of performance and the long tail, whereas here if temperature gets warmer, you may have soil moisture limited or right skewed responses. So this is also there in generally looking at soil moisture response and warming response in terms of energy limited, soil moisture limited, and so on and so forth, that you can look up. Okay, so this is for thermal performance curves describing the individual plant performance as a function of time varying temperature. If the curve is skewed, a linear response often suffices to describe variation in individual performance in response to the range of time varying temperature experienced by the plants in the wild to which they are regularly exposed and adapted. So there is the linear response versus temperature that may work in terms of at least understanding, modeling, and predictive. A negative response to time varying temperature suggests that plant performance is water limited. The red dashed line, oops, I hope I'm not, temperature limited. So here you have soil moisture limited here. Okay, so let me make sure it's not a negative response to time varying temperature suggests that plant performance is water limited. So they have so this is the negative, this is positive, so they have mislabeled, I suspect, whereas a positive response suggests that plant performance is temperature limited, so that's the red line. They have switched it up here. B to D, across a plant species geographic distribution, average growth rate often increases with average temperature. So you can see again soil moisture limited species distribution, growth rate versus temperature. So you have response on this side, and soil moisture and temperature limited species distribution. Initially there is a temperature response and then there is a soil moisture response, and it's a negatively skewed curve as well. Temperature limited species distribution, again similar skewness, but you can see compared to the slopes of the blue lines here, the slopes are reversed here, right? So looks complicated, but just try to understand the idea that plants are optimized for photosynthesis in a temperature range, light range, water availability, and then climate change affects all of them: temperature, vapor pressure deficit, soil moisture, water content, and so on. So soil moisture limitations. Okay, so across a plant species geographic distribution, average growth rate often increases with average temperature until the species thermal tolerance limit is reached. So we're looking at a solid gray curve. By contrast, the response of individual plant growth to time varying temperatures, shown as dashed lines, may be, so there is a average ecosystem curve versus individual average species curve versus individual trees curve. So let's read this again. By contrast, the response of individual plant growth to time varying temperature, so dashed lines, may be consistently negative, so water limitation, positive in cold parts of the distribution, and negative in warm parts. So you can see positive and negative, so complicated response. Whereas here in C, positive in cold parts, negative in warm parts. So temperature limitation versus water limitation. In D, consistently negative, so temperature limitation. I think they have really mangled this up, so please read it more carefully. Okay, so I'm going to leave it here and do the conclusions and future directions separately, because I want to read it more carefully. I am being a little bit brief in summarizing the main text, but there is no need to rush it. So this is already 23 minutes, so let's take a few more minutes to read the discussion more carefully. Okay, and if there is anything to clarify or something that I didn't mention properly, I will do that in the next podcast. Okay.